Namaste. Namaste. I am JV'n. I am Dr. Anupama Goyal. I am working as Associate Professor in Botany in JVWU Women's University, Jaipur. Today I will deliver my video lecture on plant breeding, which is useful for the graduate as well as postgraduate students of botany. Well, now I start with my lecture, plant breeding. Plant breeding is a science based on principles of genetics and cytogenetics. It is a science of changing the traits of plants to make them better crops and more nourishing food. Plant breeding has been practiced for thousands of years since near the beginning of human civilization. Successful commercial plant breeding concern began to be founded from the late 19th century. According to Frankel, 1968, the plant breeding is the genetic adjustment of the plants to the social, cultural, economic and technological aspects of the environment. Darwin's theory of evolution and concept of natural selection combined with Mendel's work on heredity became the foundation of plant breeding and selective breeding. Now, what is the definition of plant breeding? Different scientists gave different definitions of plant breeding according to their views. Well, according to FEM, plant breeding is the art and science of the genetic improvement of plants. Now, according to Slapper, the plant breeding is the art and science of changing the traits of plants in order to produce the desired characteristics. And according to Bernardo, what is the definition of plant breeding? Plant breeding is the genetic improvement of plants for human benefit. Now I will talk about the concept of plant breeding. In brief, plant breeding is not a new concept. All of you know it has been used since centuries, and plant breeding started with primitive people saving seed to plant in succeeding years. The concept of plant breeding applies genetic principles for producing crops that satisfy the requirements of humans. And plant breeding activities can be mainly divided into three phases. What are the three phases? First phase, establishing the genetic variation. Second phase is selection. And the third phase is test phase for selections. The principal method used by man in early days of agriculture was that of selection. So selection is also of two types. And now what is selection? Natural selection and artificial selection. So all of you are aware the selection is also of two types, natural selection as well as artificial selection. Natural selection happens naturally, but selective breeding only occurs when the humans intervene. And for this reason, selective breeding is also called as artificial selection. Now, what is natural selection? I have just told you selection is of two types, natural selection as well as artificial selection. So, I will first of all talk about the natural selection. Selection which operates in nature without human interference is called natural selection. There is no human interference in natural selection. It favors those plant characters which are necessary for the survival or adaptation of a species. The natural selection is rule in nature and it is a basic cause of evolution. So Darwin suggested that in nature the fittest ones survive. According to Darwin, in nature the fittest only survives while the rest are wiped out. So now I talk about the artificial selection, which is also called as selective breeding. Selection made by humans is called artificial selection. So the students, selection which is made by human beings or humans is called artificial selection. It is a process of selecting and propagating plants with desired characteristics and qualities from mixed populations and tries to propagate them. Selection has resulted in the development of so many varieties of plants. What plants? Such as Kalyan Sona wheat, early lemming corn. This is the early lemming corn, one of the varieties of wheat. And now I talk about the types of artificial selections. I have told you selections of two types, natural as well as artificial. Now what are the types of artificial selection? First is the pure line selection, second is the mass selection, third is the progeny selection, fourth is the directional selection, and fifth is the clonal selection. Then one by one, what is pure line selection? In this, progeny of a self-pollinated homozygous plant is obtained by selfing, and because it is obtained by selfing, it is called as pure line selection. Now what is mass selection? In this method, individual plants are selected on the basis of phenotype from a mixed population. Their seeds are bulked. There is a bulking of the seeds and are used to grow the next generation. Now what is progeny selection? In this, a selection procedure. This is a selection procedure in which superior plants are selected from a heterogeneous population on the basis of their progeny performance. And now I talk about the directional selection. In directional selection, an extreme phenotype is favored over other phenotypes, causing the allele frequency to shift over time in the direction of that phenotype. And now fifth one is the clonal selection, one of the most important type of artificial selection, a procedure of selecting superior clones from the mixed population of asexually propagated crops such as sugarcane, potato is called as clonal selection. And now I talk about the objectives of plant breeding. That was the concept of plant breeding. Now the objectives of plant breeding are to improve the yield, quality, drought, frost resistant, and to develop disease resistant crops and to develop the disease resistant crops. Plant breeding can change the genetic potential of the seeds, the farmer's raw material, thereby increasing the yield, improving quality, and enhancing resistance to disease and other natural varieties with better characteristics. Specific objectives of the plant breeding, they vary greatly depending on the crop. So some of the objectives of plant breeding are as follows. First objective is plant breeding for disease resistance. Why plant breeding is done? It is done because to develop disease resistance in plants, crop plants are attacked by various diseases and insects and pests, and it results in considerable yield losses. So when there are yield losses, in such situation the crops are made disease resistant and food production is increased, and the use of fungicides and bactericides and bactericides would also be reduced. So genetic resistance is the cheapest and the best method of minimizing such losses. Before breeding, it is important to know the causal organisms or the causative organisms and the mode of transmission. Some fungal diseases are rust, which is caused by one of the fungus called as Puccinia, and another is red rot of sugarcane, this disease, brown rust of wheat, and late blight of potato. All these diseases, late blight of potato, red rot of sugarcane, and the rust disease, are caused by various fungal pathogens, and there are some important bacterial diseases like black rot of crucifers and viral diseases such as tobacco mosaic disease. And now the cultivation of the rust resistant varieties of wheat has been attended with spectacular benefits. One of the example of wheat is Sharbati Sona. This wheat, इसका नाम क्या है? शरबती सोना of Madhya Pradesh, is rust resistant, and some of the improved varieties of wheat are C250, C253 and C228, C228. They are the wheat varieties that are highly resistant to yellow rust. So what are the important wheat varieties? Sharbati wheat of Madhya Pradesh, C250, C253 and C228. Now I tell you about the next slide, which I can show you about the types of artificial selection. Improved quality. Quality of plants produced is another important objective in plant breeding. Quality characters vary from one crop to another crop. Many of the garden plants are prized for their size, shape, density, bright color and color patterns of the flowers. And now I show you the diagrams. First of all, this diagram shows the selection, and it shows the riots bulk. The bulk is a mixture of types and can be sorted out. Single plants are selected. As you see in this diagram or this picture, the single plants, when they are selected, they are shown individually and they are tested for purity. The promising ones are later tested for yield in bigger plots using the local types as standard for comparison. Then the yield trials are conducted in the rice fields, and then last improved selection is released as a strain from the research station. And now you see this is the diagram of picture of artificial selection. It is the common mutant variety of mustard, Brassica oleracea, in which artificial selection has been done, and you see selection for flower buds, selection for terminal buds, selection for roots. So we get the broccoli when there is selection. Thank you for hearing my lecture. Thank you so much.